Hello there, ladies and gentlemen. My name is Martin Grootveld. I'm actually from Leicester School of Pharmacy, which is in the dead centre of England, and it's actually about 110 miles or so north of London. So it's quite easy to get to London and other places from there. It's an old school of pharmacy. It's been running for more than 110 years. And we do a lot of research there. I'm basically a chemical pathologist, and I do work on mostly looking into people's biological fluids to see what's wrong with them, and measuring lots of molecules simultaneously to work out patterns of metabolites which might be abnormal in certain disease conditions. And I also did a lot of work on food toxicology, and one of my favorite subjects, which I'm happy to share with you today, is on thermally stressed culinary oils, which people use a lot for frying and cooking purposes. And the title of my talk is actually Warning: Thermally Stressed Dietary Polyunsaturated Fatty Acids Are Damaging to Health. Indeed they are. Oils, as we know them, contain very, very high amounts of polyunsaturated fatty acids, or at least a lot of the oils which we deal with do. And these are the ones which people generally use for frying purposes. And they tend to be encouraged to do this by various health service and governmental regulations in their country. So I'm going to talk about which oils are dangerous. I'm going to talk about what sort of toxic compounds are actually generated from decomposition, or should I say the oxidative degradation of polyunsaturated fatty acids present in these oils. And I'm also going to talk about potential solutions which are offered by oils which contain high levels of monounsaturated fatty acids, which I've abbreviated MUFA here for you, and also saturated fatty acids too. Commonly, people will buy these oils in supermarkets or retail stores, and they will actually use them for high temperature frying episodes. And the temperature of 180 degrees centigrade standard frying temperatures, or although there are some operations which can use lower temperatures. But if we look at Chinese wok cooking, for example, the temperature can go up to about as high as 250 degrees centigrade. And in the presence of atmospheric oxygen, molecules present in these oils, particularly polyunsaturated fatty acids, are actually not stable. They degrade to very, very high levels. And we generate very, very high levels of what we call lipid oxidation products, which I abbreviate LOPS, L-O-P-S. And these lipid oxidation products get absorbed in the foods which we are frying in the oils to a certain extent. Typical potato chip, shown in this picture here, can contain anything up to, say, from 5 to 35% of its weight of the fat or the oil in which it was actually fried. And that's a very, very high level. And of course, any toxins which are generated in the oil are actually transferred to the food. And we eat food, and these toxins are then have the ability to cause some adverse health effects in our bodies. Here's another picture of oils. There's lots and lots of bottles here. The other thing people tend to do, as well as use these oils for high temperature frying episodes, they store them in the presence of sunlight, which you should not do, because if you do that, then the similar sort of reaction also occurs as well. We also get oxidation of unsaturated fatty acids. And that's why some manufacturers and other people add molecules known as antioxidants to the oils in order to preserve them. But unfortunately, antioxidants are not terribly effective at high temperature frying episodes. In fact, they tend to degrade and they decompose. Just so that you're aware, here's just a simple picture on cancer and its sources. Where does it come from? Well, apart from 5 to 10% of cancers which are genetically based, 90 to 95% comes from our exposure to environmental toxins. And basically, the diet is responsible for 30 to 35% of those cancers. This comes from a very reliable publication. Tobacco smoking, as we might expect, 25 to 30%. Infections, 15 to 20%. Obesity, 10 to 20%. Others, 10 to 15%. And cancers which have an alcohol source range from 4 to 6%. So we have a lot of diet health controversies. What is it? Is it saturated fatty acids? Is it monounsaturated fatty acids? Or is it polyunsaturated fatty acids which play roles in these diseases? Well, first of all, cardiovascular disease, and we've shown, for example, that some of the compounds which are actually generated during the frying of an oil at a standard frying temperature of 180 or so degrees centigrade, actually the same as those which cause modifications to a molecule known as low-density lipoprotein, which promotes, when it's been modified in its oxidative form, cardiovascular diseases. Some of the molecules generated, or should I really say a lot of the molecules generated from the lipid oxidation scheme, are actually very, very highly chemically reactive. And they can react with DNA. And so they can give rise to mutagenic effects and also cancers. There's a lot of influence on oils, which we use in the diet, and lipid oxidation products too. A lot of influence on the development and progression of diabetes, and also neurodegenerative diseases, just as an example. A lot of the problems which arose from people associating different classes of foods, oils and fats, sugars, carbohydrates, whatever else, with various diseases comes from something I call data dredging. And that's basically a consequence of the availability of huge databases and cheap computing power. So, for example, if we take 100 different food items, be they lipids or carbohydrates or fiber or other material present typically in our diet, and personal habits, and compare them with 100 different diseases, and then perhaps we might divide and subdivide those diseases according to the organs which they affect, we end up with a matrix of 100 times 100 cells, which is 10,000 cells. To allow for a type 1 area, statisticians, when we analyze these data, you normally actually set what's known as a probability value, and that's the probability that there is actually no association between those particular diets and those particular diseases. And if we set it at a value of 0.01, okay, that's the probability of a type 1 error, we still obtain 100 stunning significant correlations between the intake of certain types of food and basically selected diseases, even if there wasn't one in the first place. So I'm just going to talk to you about the molecular structures of fatty acids. Here's a diagram. It tells you what the structure of the fatty acids are. And you can see that we've got basically lots of carbon atoms in these chains. Lots of carbon atoms in the chains. They're actually very, very long chains. And these wiggly lines here, they refer to carbon atoms with 2 hydrogens bonded to them, apart from the one at the far end, which is a carbon with 3 hydrogen atoms bonded to it. And the polar part of the molecule is actually a carboxylate group. And because there's no carbon-carbon double bonds in this molecule at the top, steric acid, it's known as a saturated fatty acid, okay? So according to lots of health authorities, it's one of the bad guys in our diet, okay, because it's a saturated fatty acid. However, if we introduce a double bond into the chain, as we have done, sorry about that, in this molecule here, oleic acid, and it's in the 9 position, and it's called an omega-9 fatty acid because the first double bond is located 9 carbon atoms away from the methyl function at the end of the chain, then basically that's an unsaturated fatty acid. But because it's only got 1 double bond in it, it's not known as a monounsaturated fatty acid. Likewise, the third fatty acid down is known as linoleic acid, and that has 18 carbon atoms long. Again, it's got the carboxylate function, the carboxyl function at the end of the chain. It's got 2 double bonds. And it's particularly these polyunsaturated fatty acids, as it is known, otherwise known as a polyene, which are particularly susceptible to oxidation because the carbon-hydrogen bond strength in the CH2 function here is actually particularly weak. And so one of the hydrogens can actually be abstracted quite easily. Likewise, we have linolenic acid, which has got 3 carbon-carbon double bonds. And because it's got 3 carbon-carbon double bonds, it's more susceptible to oxidation than both linoleic acid and oleic acid. Whereas we find in fish oils, we've got a molecule known as eicosapentaenoic acid, which is 20 carbon atoms long with a carboxyl function right at the end, but it's actually got 5 carbon-carbon double bonds. And because of that, there's 1, 2, 3, 4 bisallylic, as they are known, CH2 functions, which have weak carbon-hydrogen bonds. So those bonds, those chemical bonds are easily breakable. And lastly, we have docosahexaenoic acid, which is based at the end. It's actually 22 carbon atoms long, but it has 6 carbon-carbon double bonds. So it's even more reactive than eicosapentaenoic acid. And we have CH2 functions here, 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 and here. And basically, the carbon-hydrogen bond strength is actually very, very weak. And this molecule has actually got 5 of those, otherwise known as bisallylic methylene functions with hydrogen atoms. Hydrogen atoms can easily come off and be degraded. So if we use any of these molecules in a frying oil, what we find is that the molecule which is the most stable is actually stearic acid. Why? Because it doesn't have any carbon-carbon double bonds. Oleic acid is also quite stable because it's only got 1 carbon-carbon double bond, but having said that, it's actually a lot more reactive towards this oxidation reaction, which happens at high temperatures in the presence of light and also in the presence of atmospheric oxygen. It's more reactive than stearic acid. Linoleic acid is a lot more reactive. It's got 2 carbon-carbon double bonds. Linolenic acid, even more reactive with 3 carbon-carbon double bonds, and all the way to eicosapentaenoic acid, more reactive, and docosahexaenoic acid is more reactive than EPA. Now, that's not the way the structure is in our bodies. Most of the lipids in our bodies have these fatty acid structures which are bonded to what's known as a glycerol backbone. And you can see this part of the molecule right at the end here. It's actually an alcohol with 3 OH functions, and that's known as a glycerol molecule. And it bonds to fatty acids, and each of its 3 positions. When these bonds hydrolyze, and these bonds are known as ester bonds, when they hydrolyze, we end up with free fatty acids. And I'm sure a lot of you are aware that we monitor free fatty acids as a measure of quality of oils, because oils containing triacylglycerols actually hydrolyze, and they generate free fatty acids, and in the end, they can also generate glycerol as well. But we get various mixtures of glycerol backbones with 1, 2, or 3 fatty acids bonded to them in this manner. And you'd also probably have a look as well. You can see that the carbon-carbon double bond here is in a particular configuration, and it's known as a cis configuration. There's another configuration known as a trans configuration, which means that the fatty acid is a different salt. And trans fatty acids, you probably know the story behind that, generated from various industrial processing techniques, have caused a lot of problems regarding health. But that's nothing compared to the degradation of polyunsaturates to lipid oxidation products. It's because of this kink that oleic acid can remain a liquid at room temperature, whereas if we actually look at saturated fatty acids, if we buy some pork fat or lard, for example, we know it's solid at room temperature. It will melt at 37 degrees centigrade, which is our body temperature, thank goodness. And it stays solid because the fatty acid chains can pack down on each other so much more easily than molecules such as oleic acid with a carbon-carbon double bond in the cis configuration. And that's the reason why, just going back one slide, fish oil contains large amounts of eicosapentaenoic acid and also docosahexaenoic acid, abbreviated DHA, quite simply because they remain liquid at very, very low temperatures. And of course, fish, as we know them, they have to survive very, very low subarctic temperatures in order to survive. Okay, here's a lipid peroxidation scheme. And this is actually a linoleal glycerol acyl chain, which means it's a linoleic, part of a linoleic acid molecule, which is bonded to a glycerol backbone. Here's carbon-hydrogen bonds. Here's a carbon-hydrogen bonds in this part of the molecule. In the bisallylic position, the ones which are actually susceptible to removal, and in the presence of heat and light, delta term represents heat, HV represents light, we get an abstraction of a hydrogen atom, we form what's known as a pentadienyl carbon-centered radical, which then reacts with oxygen. You have to fry things in the presence of oxygen, although it has been known people to develop frying techniques which don't actually involve oxygen, and they can do it under a vacuum or in the presence of nitrogen, in which case we can suppress the lipid oxidation reaction. But anyway, we form this carbon-centered radical, which then reacts with an adjacent fatty acid molecule after adding on oxygen, which reacts with oxygen to form a conjugated hydroperoxy diene species, otherwise known as a CHPD. However, the radical which is formed, which is known as a peroxyl radical, then reacts with an adjacent polyunsaturated fatty acid molecule. And remember, we might have a frying vessel packed full of lots and lots of these molecules together. All these molecules are going to have very, very close next door neighbors. And another hydrogen atom is actually abstracted. And so what's known as an autocatalytic chain reaction. In other words, it goes on and on and on until it's stopped. And it's molecules such as antioxidants, for example, vitamin E, which can prevent that reaction by terminating the process. And in that process, the vitamin E molecule can donate a hydrogen atom to stop the reaction going on. And all these reactions contain unpaired electrons. In other words, these species are known as free radicals. Now, the conjugated hydroperoxy diene molecules, which I'm indicating here, are not terribly stable, especially not at frying temperature, and they tend to decompose. They decompose to lots and lots of smaller molecules. In other words, what we say is they fragment, and they generate molecules which have these structures here. Some of the most popular or most frequent lipid oxidation products which we find are known as aldehydes. We have two different sorts of aldehydes. We have unsaturated aldehydes, which have carbon-carbon double bonds. And we have saturated aldehydes, such as this one here, N-nonenal, with 9 carbon atoms in total. The aldehydes with 2 or more double bonds only come from the more highly unsaturated fatty acids. So they come from, for example, linoleic acid, whereas the ones which only have 1 carbon-carbon double bond can come from both monounsaturated fatty acids and polyunsaturated fatty acids. So we generate these aldehydes. Now the problem is, aldehydes have this chemically reactive function right at the end, known as an aldehyde group. And it's that aldehyde group, because it's chemically reactive, which renders these molecules very, very toxic indeed. So here's some toxicological actions of those aldehydes, which arise from the degradation of polyunsaturates. They're atherogenic, they're mutagenic and carcinogenic because they're DNA reactive. They're gastropathic. Tiny levels, very, very small levels, can induce gastric ulcers in humans. They're pro-inflammatory. They have involvement in a lot of inflammatory conditions. They cause congenital malformations. And they're also teratogenic. One experiment I did with some collaborators in Spain about 20 years ago proved that. So here's some publicity we had. This publicity is quite old now, I think. This goes back about 4 or 5 years ago. This is us doing, well, one of my ex-PhD students now doing research, was doing research on an oil, which is very, very good. Got about 30 publications. Here's a recording we did for a program on our UK Channel 4 program known as Food Unwrapped, and I had to actually cook some chips in a mobile chippy van on Brighton Beach of all places. So if you like, it was live on-site monitoring of frying oil safety, because I had a number of calorie reactions and reagents I could use for calorie reactions, and I could do tests on the oils, which I did. And that experiment at least proved that coconut oil was very, very safe. And here's something in our well-known newspaper, The Telegraph: cooking with vegetable oil releases toxic cancer-causing chemicals. So, a recent development is that aldehydes have been found to be the dominant carcinogens in cigarette smoke. So these are exactly the same sorts of molecules which we're generating in a cooking oil. And remember, a lot of these aldehyde molecules are actually quite volatile. They have boiling points which are lower than standard frying temperature. And because of that, they tend to volatilize. And if we're cooking using an oil and we don't have correct ventilation in our kitchens, we can easily breathe them in. And there are connections between inhalation of cooking oils in selective restaurants, especially Chinese restaurants, I think, and the development of lung cancer. But anyway, this publication demonstrated that aldehydes represent dominant carcinogens present in tobacco smoke, which give rise to DNA damage and also block DNA repair in tobacco smoke carcinogenesis. And they also prevent many other smoke pro-carcinogens. I'm not going to mention the name of these because they're a bit too long. So basically, there were questions as to what exactly are the active carcinogenic agents present in cigarette smoke. And Weng et al. Seem to have proved that it was actually the aldehydes. And on the basis of those results, the authors of the report proposed that toxic aldehydes represent the dominant tobacco smoke carcinogens. And as documented in some of our studies, the aldehyde contents of a typical large serving of restaurant French fries are not very dissimilar to those available for inhalation during the smoking of a 20-25 allocation of tobacco cigarettes. Okay, what's worse, shallow or deep frying? I think a lot of people seem to think that shallow frying is a lot more healthy for us, whereas deep frying is not. Why is that? Well, deep frying, the food which we're frying has the opportunity to actually absorb a lot more oil than what we tend to get from shallow frying. But if we use shallow frying on a polyunsaturated rich culinary oil, such as sunflower oil, which hasn't been genetically altered, or for that matter, any other oil containing high levels of polyunsaturated fatty acids, such as corn oil, the surface area of the oil is actually exposed to oxygen a lot more because the surface area is actually a lot greater. Whereas in deep frying, the surface area is actually quite a bit smaller. It's only the surface of the oil which is exposed to oxygen. So in shallow frying, which isn't, there's a layer of oil which is not very deep at all usually, basically we generate a lot more lipid oxidation product than what we do using deep frying, because once the aldehyde or the toxic compounds have been formed at the surface of the oil, they actually dissipate into the bulk medium of the oil. So their overall concentration, which is number of molecules per milliliter or per kilogram for that matter, is actually quite a lot smaller than what we experience with shallow frying. And here's a color reaction test we can do. It's using a reagent, which I should abbreviate TBA, thiobarbituric acid. And on the left-hand side, I've just got some unheated corn oil, and that's the general colour of it, okay? Sort of a beigey yellow colour. Directly to the right of that, I've got heated corn oil, and you can see after heating, it's actually got a bit darker. And that's because corn oil contains a reasonable amount of vitamin E, and the vitamin E, was known as alpha tocopherol, actually becomes oxidized. I've got a colour reagent. This colour reagent, abbreviated TBA, or otherwise known as thiobarbituric acid, is actually reacts with aldehydes to form a bright red coloration, bright pinky red coloration, to be honest. And this is me adding some of the thiobarbituric acid reagent to unheated corn oil in the third beaker along, or should I say the third vessel along. And we can see there that there's not much change in color at all, okay? And that's without heat, that's actually without heating the oil. You know, there's the arrow there, so you can see. Now, this is me doing the same thing with the corn oil, but after heating it at 180 degrees centigrade for a period of an hour, okay? And look at the intensity of the color. That's just reflecting a very, very large quantity of toxic aldehydes, which I'm actually generated in that polyunsaturated rich culinary oil. So we can see that using a simple color reaction, we can easily demonstrate very, very high levels of aldehydes. If this was a monounsaturated rich oil, such as olive oil, rather than corn oil, which is high in polyunsaturates, the color intensity won't be quite so good. What's the best way of analyzing for these molecules? Well, we use a technique known as high resolution NMR. NMR stands for nuclear magnetic resonance analysis. And it takes advantages of the magnetic properties of nuclei or certain atoms which are present in molecules. Unfortunately, hydrogen atoms are actually magnetically reactive, and they're ubiquitous both in living systems and also in lots of food samples as well. And all of them have different chemical and therefore magnetic environments in molecules. So all different hydrogen nuclei present in particular molecules give signals at different frequencies. So we employ energy from the radio frequency region of the electromagnetic spectrum, was known as radio waves, to detect changes in the alignment of nuclei when placed in an external magnetic field. And when we use the best machines around, which are very, very high operating frequency, which we have actually got in Leicester, you see the magnet's actually quite big, and this is actually a 600 megahertz magnet. That's what's known as the operating frequency. And we use the energy from the radio frequency to change the alignment of those nuclei. So when the nuclei are in the lower energy state, they align themselves with the external magnetic field, but then we excite them with particular radio frequency, which is of a certain energy. It's characteristic of that hydrogen atom present in molecules. The nuclei become aligned against the film, so they get excited to a higher energy state. Basically, the setup, we have a computer, we have what's known as a console, otherwise known as a spectrometer, transmits and receives the radio frequency waves used to make the NMR measurements, and we have the superconducting magnet. So it generates a powerful magnetic field that's tens of thousands of times stronger than the Earth's magnetic field. And samples are placed within that magnetic field and exposed to radio waves. We use basically an NMR tube, which is about 7 inches long and has 5 millimeters diameter. We usually use about half a mil of oil for analysis, or half, you might use it for looking at a biological fluid, such as a urine sample or a saliva sample. And we put about only half a mil or so into an NMR tube. And the sample is then transported from the top of the magnet, but usually via an autosampler, and we've got an autosampler, and decreases slowly to the bottom of the magnet, where we have what's known as a probe. which controls the radio frequency transmission to the sample. And there's an old publication of mine from about 20 years ago, and it's on human saliva. I just want you to appreciate all the signals we get in a typical NMR spectrum. This is just one NMR spectrum, and I believe it's my saliva. Can't be sure about that. So you can see all the things that I was getting up to. We generally collect saliva samples first thing in the morning, so we have no contamination from people's diets or toothbrushing or other things. So we get them to collect a sample before they do anything else with their mouths. And we get a series of signals, and typical signals we might find in saliva are due to molecules which are actually generated from bacteria, or otherwise known as the oral microbiome. But in order to interpret an NMR spectrum, we can look at the structure of a molecule, and here's a molecule known as propionate, and I've got a methyl function here and a CH2 function here. And the more shielded that a nucleus is from the magnetic field, then basically the lower the frequency. This is a frequency scale, and we measure it from right to left as opposed to left to right, which is what we do with some other spectroscopic techniques. So here's actually the high field region of the spectrum. Here's the middle field region. Here's the low field region here. Likewise, this is low frequency, and this is high frequency, because frequency is inversely proportional to field strength of the magnet. And basically, this molecule propionate, you can see it's got two different types of hydrogen nucleus. Well, one of them has got a signal here at this particular frequency, which is about 1.05 of a part per million. These field strengths are invariant according to spectrometer strength. We have another signal here at about 2.15 parts per million for the other CH2 signal. This CH2 signal is less deshielded, is more deshielded, I should say, than the methyl function here. It's because the methyl function is a long way away from the oxygens in the carboxylate group, which are electronegative elements, that we get the shielding and deshielding. Likewise with the pyruvate molecule, there's only one methyl function, so only one signal, and that comes at, if my memory serves me correct, at about 2.38 parts per million. The PYR is the abbreviation for pyruvate. Well, what have I been doing? I can see some ethanol here, and turns out I had a drink the night before. I did actually drink some alcohol, so I can see some ethanol there. I've got molecules here due to compounds known as methylamines, which are actually quite smelly, but everyone's got them, so I really needn't worry about that at all. And for smokers, I can actually often find high levels of a compound known as methanol, which is actually quite toxic because it's metabolized to an aldehyde, which is formaldehyde. And methanol comes from the combustion of tobacco lignin. Well, let's move on to an oil. We can look at the aldehyde region of oils which have been thermally stressed, and we can use this NMR technique. And here's some spectra, as they're known. And it's only a small region. It's a 9.4 to 10.2 part per million region of the spectrum, which shows us the magnetic environment and signals resulting from the analysis of aldehydic function regions, CHOs. And you can see I can detect about 9 different classes of aldehyde in a sample of canola oil, which has been thermally stressed from 0 up to 90 minutes. And the more we heat, and this is at all of the temperature of 180 degrees centigrade, the longer we thermally stress for, the higher the concentration of the aldehydes which are generated. And you can even see that even after 5 or 10 minutes, we start to see some signals starting to develop. What are the advantages of the technique? Well, it's virtually non-invasive. It's multi-component. It can analyze lots and lots of molecules simultaneously. So if I can take an oil sample and I can not only tell you what sorts of fatty acids you have in that particular oil, I can tell you about the major components, and I can also tell you about the minor components, such as lipid oxidation products. It's valuable for the multi-component monitoring of a very wide range of lipids and lipid oxidation products in oils and also food samples. We can do analysis on potato chip samples if we wish to. And it provides simultaneous multi-component information regarding the chemical nature and levels of lipid oxidation products generated in those oils. There's only a minimal sample preparation, fast analysis time of about less than 10 minutes or so for an oil, and therefore we get rapid sample throughput. And the other advantage of the technique is that we can identify molecules which we would not normally anticipate to be present in those samples. For example, new, previously unidentified lipid oxidation products. By the way, when I mention LOPs, that's an abbreviation for lipid oxidation products. Well, what are the concentrations of LOPs generated during frying episodes? Conjugated hydroperoxidines up to about 60 millimoles per kilogram. Some of you who did chemistry or remember doing chemistry at school, some of you who have done it to a higher level will know that 60 millimoles per kilogram is actually quite a high level compared to the concentrations we often experience of other toxins. But the conjugated hydroperoxidines, which fortunately fragment at frying temperature to unfortunately aldehydes. We have saturated aldehydes and compounds known as alpha beta unsaturated aldehydes. And it's the alpha beta unsaturated aldehydes, which are generally a lot more toxic than saturated aldehydes. And we get very, very high levels of those. And I've got these some typical figures we might get in millimoles per kilogram. And the other thing you have to remember is that these are actually only residual levels. And many of the aldehydes which have been generated have a high level of volatility at a standard frying temperature of 180 degrees centigrade. So we only measure in the oil what we've lost into the atmosphere or the environment. And remember, if you're walking past a frying pan containing frying chips, deep frying or shallow frying, whatever, although it's worse with shallow frying, as I've said before already, you'll be breathing in those compounds. Here's some corn oil, which we've taken from purchased from a supermarket or a retail market, I guess you might like to call it. And you can see that even at 0 minutes, we can detect some of these aldehydes. 30 minutes, quite a lot more. 30 minutes of thermal stressing. And this is after 90 minutes, we get very, very high levels. And this is what's known as a heat map, showing increasing levels of aldehydes generated in different sorts of oils. So we can see that we use canola oil, which is actually quite high in monounsaturates, actually quite high in monounsaturates. I beg your pardon, this is canola oil. We can see at high concentrations, deep red color indicates high concentrations of aldehydes at a 90-minute time point. This is coconut oil, which is very, very high in saturated fatty acids, 90% or so. Very, very little aldehydes generated because the amount of polyunsaturates and monounsaturates is actually very, very small. Corn oil, we also generate quite a lot of aldehyde, as you can see at the high times of heating these oils. And likewise, sunflower oil, which is also very, very high in polyunsaturates, also generates very, very high levels. Here's some plots of alka 2,4-dienols, which are basically unsaturated fatty acids, which only come from oils which contain high amounts of linoleic acid or linoleal glycerols, as they're otherwise known. You can see in sunflower oil and canola oil, we actually get quite a lot, whereas extra virgin olive oil and coconut oil, we actually get very small amounts. Okay, here's some sunflower oil, which only heated for 0, 5, and 10 minutes. And again, these are the patterns of aldehydes we get. So it just goes to show you can actually detect them in oils which haven't been subject to heating. And that's presumably because they've been, during their transport and storage by retailers, they've been exposed to light. So we now come to estimating the human dietary intake of lipid oxidation products. I just want to give you, if you like, a hypothetical example, first of all. One average 154 gram serving of French fries contains 5 to 30% weight against weight frying oil lipid, okay? And let's assume it contains 15%, somewhere in the middle between the 5 and 30% weight against weight content, with an assumption of only 10 millimoles per kilogram total aldehydes. And that concentration is actually quite conservative. It's actually quite small compared to the amounts we normally get. If we do thermally stress the oil quite a lot, which simulates a reuse of the material by a typical fast food, a typical fast food outlet, then we're going to end up with what's known as 0.23 of a millimole of aldehydes in 154 grams of French fry portion. And the World Health Authority's tolerable daily intake level for acrylamide, and these tolerable daily intake levels for these aldehydes, they're not terribly popular, they're difficult to find, and there's not that many aldehydes which actually have them, but acrylamide is one of them. It's the smallest, but albeit one of the more reactive alpha beta unsaturated aldehydes available. Is actually 7.5 micrograms, which is 0.13 of a micromole per kilogram of body weight per day. Which means an average human can be exposed to 525 micrograms of that particular aldehyde per day for an average 70 kilogram adult. So if all of this 0.23 of a millimole, as calculated here, of aldehydes was actually acrylamide, it would be equivalent to 12.9 milligrams as opposed to 0.525 milligram limit. And that's only 1 meal portion. Remember, lots of people with potato chips maybe twice a day. Not everyone, I agree. Some people choose to do that, some people choose not to. And the other thing we have to remember is that more than 70% of the aldehydes generated generally are the much more toxic alpha beta unsaturated aldehydes. Here's a sample of French fries again, typical French fries purchased from a fast food outlet, and we've got different classes of aldehydes detectable in spectrum A. In spectrum B, we've just bought some packaged potato chips. I think you Americans call them chips, we call them crisps. And again, we've got a series of aldehydes which are detectable in spectrum B as well. So it's not surprising, really. They use a lot of polyunsaturated fatty acids in their preparation. The other thing I want you to know is that basically the amount of aldehyde we get in potato chips is proportional to the amount of lipid which is actually absorbed by those potato chips, okay? There are ways and means of actually blocking the uptake of cooking oils into fried foods, which we haven't got time to cover at the moment. But this is the total lipid content in moles per kilogram of potato chip samples, which we purchased from quite a few fast food restaurants. And here's the aldehyde content in figures known as micromoles per kilogram. And you can see as the total lipid content increases, so does the amount of aldehyde present. The relationships are not great. We wouldn't expect them to be great because they're going to be dependent on so many different parameters, such as frying oil reuse, frying oil time. But there are some vaguely linear relationships between them. And the red curve is actually trans 2 alkenyls. The blue curve, or straight line, I should say, are trans alkyl 2,4 dienals, which only come from the pro-oxidation of polyunsaturated fatty acids. And in green, we have compounds known as N-alkanils, which are perhaps more stable saturated aldehydes. So the estimated aldehyde masses, milligram per potato chip serving for small large and super large servings of potato chips is actually shown here. And I've got another health regulation, this time from the Australian Government Department of Health, specified the acceptable daily intake of acrolein. As I said before, it's the simplest, although similarly toxic, alpha beta unsaturated aldehyde, specifically that which is considered to be a level of intake of that molecule that can be ingested over an entire lifetime without any appreciable risk to health, to be only 0.5 of a microgram per kilogram of body weight. So that's a grand total of only 35 micrograms for an assumed average human body weight of 70 kilograms. I know that this body weight can be lower in some parts of the world, or this average body weight can be lower in some parts of the world than it is in others, but we're just using the figure of 70 kilogram average body weight at the moment. And these are the most popular aldehydes which are generated, should I say the most frequent aldehydes we find generated from the lipid oxidation process of either oleoyl glycerols, which as you remember peroxidize a lot more slowly than linoleal glycerols, which have two carbon-carbon double bonds, which in turn peroxidize even more slowly than linolenyl glycerols, which is an omega-3 fatty acid, which is present in soybean oil at a level of about 7 or 8%. And these are different serving sizes: 171 grams, 154 grams, 400 grams. You might be surprised at that, but people do eat as much as 400 grams sometimes in servings of French fries. And if we take the 154 gram serving, which is typical large size or defined as a large size for a fast food restaurant, you can see that the quantities of aldehydes, which we've determined here, ranging from about 1.5 to 2.5 milligrams, are a lot more than the 35 microgram limit set by the Australian Government Department of Health, and that's for an assumed average human body weight of 70 kilograms. We also have acceptable daily intake values for acrolein, some other aldehydes, and also acrylamide and the weed killer paraquat in terms of milligrams per kilogram of body weight per day. And you see the figure for acrolein, 0.0005 of a milligram per kilogram of body weight per day, is actually nearly 10 times lower than that for paraquat, which means it's nearly 10 times as toxic as paraquat. And the paraquat value I've got here for the acceptable daily intake is 0.004 of a milligram per kilogram of body weight per day, whereas that for acrolein is 0.0005. So if I'm correct in saying that means acrolein is 10 times worse. I've also got figures for acrylamide. Acrylamide got a lot of publicity. It still has publicity now on how bad and toxic it is. It's actually it's possibly carcinogenic, and it's a fertility toxin. And basically, the acceptable daily intake for acrylamide is actually a lot greater than what it is for acrolein, with paraquat somewhere in the middle. So acrolein represents the simplest, lowest homolog trans-2-alkanone class of aldehydes and is conceivably one of the most toxic of those lipid oxidation products. One estimate dating back to 2001 was only 70 micrograms a day, but a more recent and more reliable estimate, the mean dietary acrolein intake from a total of only 8 foods, was as much as 2.35 milligrams per day, which is equivalent to 34 micrograms per kilogram for a 70 kilogram body weight human. And that value is 33-fold greater than the above likely underestimate made here. Moreover, the maximal level of this aldehyde's dietary exposure was estimated as much as 5 milligrams per day. And down here in red, these are other people's data. These are estimates made by other researchers. Human exposure levels for total aldehydes have been estimated to be as much as 7 milligrams per kilogram per day, with 5 milligrams per kilogram per day representing the more reactive and toxic alpha beta unsaturated aldehydes. And here's a little bit about acrylamide. Here's chemical structure here. You can see it's actually quite similar to that of acrolein. It's just that acrolein has a CHO function here, which is an aldehyde function. Acrylamide has an amide function, CONH2. Exerts toxic effects on the nervous system and fertility, and may also be carcinogenic. Its absolute maximum level in fried foods is only 4 parts per million, which represents an equivalent level of 56 micromoles per kilogram of potatoes. And interestingly, there is much evidence to suggest that acrylamide may arise from an acrolein source when asparagine-rich foods are fried in polyunsaturated-rich culinary oils. Here's something on the mutagenicity of Chinese cooking oils. This is going back quite a long way, 20 years or so. Volatile emissions from heated culinary oils utilized for the purpose of Chinese style, perhaps including wok cooking, are mutagenic. And researchers, some of them from the National Institute of Health, suggested that such indoor air pollution may render humans susceptible to an increased risk of lung cancer. And acrolein, extremely toxic trans-2 alkyl, as we've said before, was present in those fumes. Well, why are these compounds so toxic? Well, this aldehyde function could react with amino functions present in amino acids present in proteins, also react with amino functions present in DNA. And that's why we can structurally modify DNA. And that such processes, if perpetuated, can give rise to cancer. They can give rise to a lot of other conditions as well. But this is known as a shift-based product. And there's also a Michael addition reaction as well, which can involve a thiol group, such as that in the amino acid cysteine or the tripeptide glutathione, or another amino group too, where basically there's reactivity of the thiol group at the C3 position, or the nitrogen in the amino function, also at the C3 position in these particular alpha beta unsaturated aldehydes, and we form these derivatives here. Okay, here's a little bit on atherosclerosis. This is what a normal artery looks like. So it's quite clear. This is diseased artery where it's been narrowed, and this is due to the presence of fatty streaks within the artery. And here's a blocked artery, also a disease state, arising from high infiltration of fatty streaks. And here's our paper from 1998, confirmed that basically aldehydes from the diet, we fed these to experimental animals, basically were actually absorbed from the gut into the systemic circulation and were excreted as mercapturate conjugates. So actually proving that we found aldehyde metabolites in the urine of these animals proved that basically if they were ingested orally, then basically they were actually absorbed. So we came up with a scheme in our Journal of Clinical Investigation paper whereby people eat dietary polyunsaturates or use dietary polyunsaturated fatty acids for frying purposes. There's an oxidative modification in the presence of oxygen and also heat or light. We form the conjugated diene hydroperoxide species, which fragment at high temperature. Like I said, these species here, they're incredibly toxic themselves. So it's lucky that they fragment, but they fragment compounds which are almost as toxic, known as aldehydes. Having said that, we still actually find significant levels of these conjugated diene hydroperoxides, otherwise known as, you might know them as lipid peroxides or lipid hydroperoxides, present in heated culinary oils, especially those that contain high levels of polyunsaturates. So we form these saturated and alpha beta unsaturated aldehydes, we eat them, they're absorbed from the gut into the systemic circulation, whereby they react with a molecule known as low-density lipoprotein, and they react with a protein component of low-density lipoprotein, which is known as the ApoB part. And there are particular amino acids containing side chain amino functions, there's lysine residues present in the ApoB part of the low density lipoprotein molecule, so it changes the structure. Once the structure has been changed, that particular form of low density lipoprotein, which is actually abbreviated LDL, becomes recognized by scavenger receptors present on cells called macrophages. And so the macrophages actually take up large amounts of fat, which is also associated with low density lipoprotein. That's why it's called low density lipoprotein. It actually transports fats and lipids and also cholesterol. So they're uptaken by macrophages, and we form foam cells which can conglomerate together to form fatty streaks which block arteries. Here's our experiment we did. I don't want to go into too much detail. This is top spectrum, is a control rat urine NMR spectrum. I can tell it's a rat urine spectrum because I've got a signal here from a molecule known as allantoin, which is a final product of purine metabolism in rats. We don't see that in human urine because our purine metabolism stops at a molecule called urate, which can have some disastrous consequences, unfortunately, because high levels of urate depositing in joints gives rise to a condition known as gout. Not to say that animals don't get gout as well, they do if they have a problem with the enzyme that converts uric acid or urate to allantoin. And you'll have to take it from me that all these signals which have been highlighted in the bottom spectrum come from this metabolite here, which is a mercapturate conjugate arising from the metabolism of the aldehyde which we gave, which is trans 2 noni now to these animals at that particular dose level. So that's proving that the aldehydes are actually absorbed from the gut into the systemic circulation, where they can exert their toxic effects and are actually metabolized and excreted in the urine. Here's a scheme for the metabolism. I'm not going into too much detail, but this is the aldehyde we start with, trans 2 noni now, sorry about that, right at the top on the left-hand side, abbreviated T2N, and the various enzymes taking part in the reactive process, and we end up with what's known as a mercapturate conjugate. Here's another fact for you. In 1978, the Demographic Yearbook of the United Nations reported that the Sri Lankan population had the lowest documented mortality rate from ischemic heart disease of all the countries surveyed. And their predominant dietary fat is coconut oil, which contains approximately 95% saturates and a very low content of polyunsaturated fatty acids. And there's some other populations in the world demonstrating this paradox. We've all heard of the Southern French paradox. People are eating large amounts of saturated fats, for example, and also the saturated fat eating Maasai tribe in Africa. Here's some further evidence for the in vivo absorption and potential pathogenicity of polyunsaturated fatty acid derived aldehydes. Diets, number one, diets containing thermally stressed polyunsaturated rich oils are clearly more atherogenic than those containing unheated oils. And that's published in the very reputable journal of Atherosclerosis Research in 1967 by Krzyzewski and Tepper. Secondly, oxidized preheated dietary polyunsaturates accelerate the development of atherosclerosis in New Zealand white rabbits. That's by my ex-colleagues, Dr. Staprans in San Francisco. 100% increase in fatty streak lesions in the aorta observed after feeding with a diet containing lipid oxidation products. And thirdly, the dietary consumption of peroxidized culinary oils enhances the accumulation of lipid oxidation products in macrophages and monocytes. Paper by Smith and Kamaru, more recent this time in 1987. Here's a study we did with a group in a very active research group in Madrid. basically, I think some of these people are still there, but it's published in journal Free Radical Research in 2002. We looked at the teratogenic properties of heated safflower oil, which is actually rich in polyunsaturated fatty acids, and we looked at the rates of yolk sac resorptions and embryo malformations, and we found that they increased strikingly following the feeding of diet supplemented with preheated safflower oil and its administration to pregnant experimental animals. And heating the oil substantially increased its concentrations of lipid oxidation products and decreased its alpha TOH, which stands for alpha tocopherol, which is vitamin E. And the extent of embryo malformations in the heated oil group was 22% compared to 5.6% in the unheated oil group. A little bit about gastropathic properties of lipid oxidation products. Oral administration of the lipid oxidation product 4-hydroxytrans-2-nonene now to rats at a dose level of only 0.26 of a micromole per litre, which is a concentration very similar to what we normally find in healthy human blood plasma. That induced peptic ulcers in those animals. And if you're interested in that, here's a reference. A little bit of my work for the United Nations. I go to Africa. I haven't been for a couple of years now because of COVID, but I visit countries such as Uganda, Kenya, and Tanzania in order to help them develop their culinary oil industry. And they got, because they're actually grown in different climate, they actually have sunflower oils, or at least they have sunflowers which generate oils which are actually quite high in monounsaturates and relatively low in polyunsaturates. So we're looking into developing these for, if you like, virgin oil use and perhaps sale in the European and perhaps USA market. Here's a typical laboratory we started to develop in Uganda back in 2018. See, it looks pretty basic, but if you're starting a new company for the manufacture of oils, there are a lot of color tests you can use, which are actually quite simple. You don't actually need much in the way of sophisticated analytical equipment. And some of these color reactions you can actually monitor on your mobile phones. So it's actually quite handy. So this is what I've advised to them, and I've helped them set them up. Also had a look at health and safety and their factories. I employ a lot of women, and a lot of these women are exposed to lots of sunflower dust in actual fact. And so we've advised that they wear masks, wear white coats, and wear gloves. Do the work to the best of our ability at very high, with very high regard for health and safety. Here's a research group meeting of us. That's me some years ago. This guy here is Victor Ruiz Rodado, who helped me with some of my oil work. He's actually doing work on metabolomics, so he's actually now working at the National Institute of Health. And here's some other people with various backgrounds who are actually working with me at the time. Here's a copy of my book. I'm not trying to plug this, but this came out in 2014. I've got another book at the moment. It's on metabolic profiling. And I'd just like to thank you for listening to my presentation. Thank you very much for that.